What I'm going to do is talk about therapies that we're doing and other people are doing for spinal muscular atrophy. But before I do that, I just wanted to sort of set the background a bit. I'm the director of the West Australian Neuroscience Research Institute, and where we're based at the Centre for Comparative Genomics at Murdoch University. Before you can come up with any therapy for any disease, you have to have an understanding of the disease mechanism. I mean, you can't fix a car if you don't know what's wrong with it. So you have to work out precisely what is wrong, so then you can come up with some rational therapies. So just to start with the West Australian Neuroscience Research Institute, this was previously the Australian Neuromuscular Research Institute. I joined this in 1991. I was involved in setting up diagnostic tests for genetic diseases, including Duchenne muscular dystrophy and other conditions. Beginning of last year, I was approached by I've been approached by Murdoch University, and I joined them and shifted campuses. Where we were based, the ANRI is still at the QE2 campus in A Block, and this is where there's some specialist clinics are. ANRI is the oldest private medical research institute in Western Australia. It's been going since about '82. And the beauty of this institute is it brings clinicians and bench researchers together on the same floor. We interact, and the idea is we try and work together. There's no competition, it's real collaboration. So on the QE2 site, we're looking at stroke, brain plasticity and restorative neurology, whereas where I'm based at Murdoch, we're looking at, again, we have the same specialist clinics, but we're developing the genetic therapies for a number of diseases. And also we're interacting with a number of biopharma to try and bring these therapies to the clinic. So I'm the director of this place now, so I'm sort of bouncing between two campuses, although I spend most of my time at Murdoch. So what we're doing is we're looking primarily at neuromuscular disorders. And on this slide, this is just some of the guys from Western Australia. The young lad here has spinal muscular atrophy. There's a couple of limb girdles here, and there are the rest are Duchenne muscular dystrophy patients. Most of the work we've been doing is on Duchenne muscular dystrophy, and this is where I met Julie in 2008, talking about how we were trying to come up with a therapy for Duchenne. That work has progressed with leaps and bounds, and it's really going to be the flagship, I hope, of this type of treatment for all sorts of neuromuscular conditions. So what I just want to do, and don't be intimidated, I'm going to be talking about basic gene expression, mutations, and that's how things go wrong, and then when you know how they've gone wrong, how we can correct them. Now, if there are any questions, don't wait until the end, just ask straight away. Don't even put your hand up. Just ask if I don't make anything clear, ask for clarification straight away. So, gene expression. Our genetic material is carried in genes. And just think of it as a genetic Morse code. We've got A, C, G and T. And it's the order of those A, C, G and T that carries all the genetic information. So think of a gene as a page in a book. And that book is like a chromosome. You've got a lot of genes on one chromosome, and you've got all these books in our library that make us up. So we're just looking at one gene. We have about 25,000 different genes, and they all go through the same sort of basic steps. They're switched on in a process called transcription, splicing, and I'll be talking about that in more detail, because in splicing, the yellow bits here are the important bits. The aqua bits, or the light blue bits, this is called the intron. This is the non-coding sequence. And in the splicing process, all these yellow bits are joined up to make a continuous message. And it's that continuous message that's translated into making a gene product or a protein. And the message is read in three-letter words. Very, very precisely three-letter words. So the message here is the bad and old dog ran and ate the fat cat. End of message. Now this is a very abbreviated version. Some of these genes, they have, what is it? About 100,000? 30,000 words in one sentence. So they're very big. And in some aspects, the bigger the gene, the more prone it is to getting spelling errors. So the step of splicing, I just want to go through in a bit of detail. So these were the yellow blocks. These are called exons. We've got the introns in between. And the average gene is 8 to 9 of these exons. And it's spread across 30,000 ACGTs. That's the average gene. And the SMN gene, it's responsible for SMA, is very average. 8 exons spread across 30 kilobases. The gene I'll be talking about for muscular dystrophy, dystrophin, is not average. It's 79 exons spread across 2.3 million ACGT. So it's a huge gene, very complicated. And so these are the sort of the steps. So here we've got our their yellow bits, and the eyes are the intronic non-coding sequence. And what the cell machinery does is when the gene is switched on, you snip out the introns and bring the message together to make the message. Is that okay so far? Now this is very simplistic, but this happens for all our genes. All our genes go through the splicing, well, not over 95% of our genes go through the splicing process. And of that, over 75% of our genes undergo a process called alternative splicing. That is, in different cells or tissues, you get an exon being retained or skipped. So the full-length message might be, the bad and old dog ran and ate the fat cat. But then through alternate splicing and the use of one exon or a couple of other exons, you can get shorter messages. So you get different gene products from the same gene. So splicing has really increased our genetic plasticity. So we have 25,000 genes, twice as many as a fruit fly. So we're a little bit more complicated than the fruit fly, and it's because of the alternate splicing. We generate more, for those who are feeling genetically challenged, we generate a lot more gene products through this process of alternate splicing. Now, think of splicing as machinery, where we've got an exon here and an exon here. And you get all this machinery assembling at specific points in the gene. And this is what it's like. This happens for all our genes. This exon is going to be joined very, very spliced or joined to this exon. All the middle sequence is snipped out very, very precisely. And this process happens for all our exons. I said we had 25,000 different genes. They've estimated there's, last I saw was we had 230,670 exons. So in the different genes. But the process of joining one exon to another or skipping it is very similar. So it's the same basic machinery. So what we're doing, what we've done with our developed for exon skipping for muscular dystrophy, is a little genetic drug that acts like a spanner that very precisely we can drop into the machinery at one point. So we know what the sequence of the gene is, we can design our drug so that it binds to this little point and it stops one of the factors from binding. If you stop that part of the machinery from binding, the whole complex falls to bits, and then this exon will then move across to an exon, to the next exon downstream. So this is a process called exon skipping. And what I'm just going to talk about now is how we've applied it to muscular dystrophy. Because this is the work that I was talking about, and then Julie approached me afterwards. Could we extend this work to SMA? So this is the most common and serious form of childhood muscle wasting. It's caused by the inactivation of the dystrophin gene. It affects boys because it's X-linked, it's on the X chromosome. So women have got an X and a Y, males have an X and a Y, so they've only got one X chromosome. Females, XX, they can carry a defective dystrophin gene, but they have a backup copy. So with muscular dystrophy, symptoms present from the ages of 1 to 5 years of age. The boys look normal to start with, and that's because the young muscle is growing fast enough to keep up with the degeneration. But they're non-ambulant by the age of 12, and death from cardiac and respiratory complications. More commonly now, it's cardiac complications, but respiratory issues are huge. I mean, we had storms in Perth 6 weeks ago, and we lost 2 young men with muscular dystrophy because their ventilators didn't have power. Now, there's a milder form of muscular dystrophy called Becker, and this is caused by a partial inactivation of the gene. It's like pulling one plug out of a V8 motor. It's still going to run, not perfectly, but it's still going to run reasonably well. You pull out 2 plugs, it'll still chug along a bit. And so you've got a spectrum of severity from very, very mild to, in some cases, they're diagnosed by accident or late in life. There's one example where a person was found to be missing a chunk of the dystrophin gene, and he was 65 years old when he was diagnosed with the disease. He'd been playing pennant-grade badminton at the age of 62. If he had a slightly different mutation in the gene, he would have been in a wheelchair by the age of 12. So what we're trying to do with our exon skipping therapy for muscular dystrophy is change a defective gene into a semi-functional gene. Now this is a guy called Pat Mushin. He's a 43-year-old now, music teacher in Boston. He originally was diagnosed with Becker muscular dystrophy, and he was sort of one of the pin-up boys. He would go out as an inspirational speaker of living with muscular dystrophy. Since the last couple of years, they actually found out he has a form of limb-girdle muscular dystrophy, but it's still muscular dystrophy. So what happens when you have inactivation of a gene, you can have a single spelling error in a gene. The mutation, here we've got our spelling error, the mutation is carried through to the mRNA, and that will stop the message from being read. So the very easy way of looking at this is you've just changed one letter. You've changed the A to an E. We've got the bad and old dog ran, end. Now imagine when you've got a gene 2.3 million bases long, and you've got what you can change one letter and have catastrophic inactivation. And this is the typical of what happens in Duchenne muscular dystrophy. You've got a protein where you need the ends of the protein. When you've got this type of mutation, it stops working. So what we're doing with our therapy is we're interfering with the splicing step. We're actually taking out this end signal during the splicing. We're removing it so that we have the bad and old dog ran, and ate the fat cat. So we've actually used almost like whiteout in the message to stop the cell machinery recognizing that. So we've got a shorter shock absorber. I'm not going to go into the details. I think videos are worth a lot more. We've designed a Band-Aid to remove the end signal in a mouse model, a severe mouse model of muscular dystrophy. Now this animal, 9 weeks of age, they live to about 12 weeks. You've got a severe kyphosis, a curvature of the spine. It's struggling for breath. It's not a very happy mouse. It'll be dead within 3 weeks. This is, oops. This is a 9-week-old littermate of this one. We've injected our drug into this mouse, and you can see it's rather active. It was treated out to 1 year of age. So we've snipped the end signal out of the gene that causes this, and we've rescued expression. Now, there's a few steps along the way, but we've designed the same sort of Band-Aids to treat human mutations. And we've got one compound that's in clinical trials in America now. Now, with muscular dystrophy, as the boys get older, they lose the ability to walk. Now, one of the ways of assessing this is called the 6-minute walk test. And what they do is they just have 2 cones up, 25 metres apart, and just measure how far the boys can walk in 6 minutes. Now, this is reasonably subjective because it depends if the boys have had a good night's sleep the night before, if they're compliant. It is subject to some manipulation. But when it's carried out consistently, it's a reasonably accurate assessment of how the boys are travelling. But what happens is, from over the age of 7, there's a progressive decline in how far they can walk. So this 0 is, say, 500 metres. And then over time, they start walking less and less. And over a 1 year period, if the decline is greater than 10%, then it's going to continue on the downward track, and the boys will become non-ambulant within a couple of years. Now, in the trial that they've done in the States, they're receiving this drug once a week, intravenously, and when you overlay how these boys are travelling, this is the continually treated group. They had 9-year-old boys in this trial, on average, 9 years of age, and this is about 400 metres, you overlay it, and over a couple of years they dropped 10 metres over that period. The gold group here was a placebo group. So for the start of the trial, for the first 6 months, they were receiving saline. And they didn't know if they were being treated or not being treated. But what happened, there was a decline was seen in the beginning of the placebo group. We could show that the protein, the missing protein, was being restored, and the drug was working the way we anticipated it was working. So the placebo boys then started to receive the drug. And it takes a while to build up. It takes at least 12 weeks for the drug to kick in and start to work. So the decline continued, but then they've stabilised. Now this is 120 weeks worth of data. It's coming up to 3 years now, and it's still stable. They've dropped a few percent, and this group is stable as well. So the things to take home from this is that we're not going to get a boy out of a wheelchair, but if we start the treatment early enough, we can probably delay him going into a wheelchair dramatically. Now that was one test. The other tests that were being done is respiratory function. The inhalation pressure, inspiratory pressure, and exhalation. How much you can breathe in, the pressure to breathe in, the pressure to breathe out. And as the boys get weaker, the breathing gets harder and the breathing declines. So these are published studies of untreated, the natural history of the disease. And this will be the same with SMA to a point. But with muscular dystrophy, it's relentlessly progressive. There's a continual decline. The boys on the trial have been stable. If anything, they've improved a few percent. So that's looking very, very encouraging. And again, the videos, I'm lucky in the fact that one of the mothers of the boys keeps sending me updates on how Billy's tracking. Now, this was taken over a year ago, and according to Terry, whoops, he would not walk down this path unless he had a parent on either side of him. This was a year beforehand. So he's just yelling up and going down the hill, and he just sort of wanders off. Maybe I should have used sound, but it doesn't matter. You can hear a dog barking in the background. But what's more impressive is this one, where he's coming up the hill. Now, he's still on steroids. He's still got the Cushingoid features. He's tail walking because of tendon contracture. But he's charging up the hill. And I'm not sure if you'll be able to hear it. He's whistling. So the breathing is really quite dramatic. You can see the toe walking, but he was whistling. So this is again supporting the previous studies that the breathing has improved. I should point out that 2 of the boys in this trial went off their feet very early in the study. They were the boys that just qualified. They could only just walk 250 metres, and they basically became non-ambulant before the levels of dystrophin became sufficient to make a difference. However, they're still in the trial, they're still receiving the drug, their breathing is fine. So it's been stable. And then another couple of... This is that's not Billy, but he's coming up here. Now this is, I don't know if I'd get around this, it's quite a circuit. And at the end of it, he's going to do a wheel shunt or whatever that is. So, again, it's supposed to have, he's about 12 years old at this stage. And this is him from a couple of weeks ago. 13 and a half.
Oh, yeah.
A couple of months ago, he actually finished the Pittsburgh Children's Marathon. He walked most of it and then jogged across the line. And his mum asked him, why were you jogging across the line? He said, I was watching the clock. I didn't want it to tick over another minute. And then he was complaining that they weren't playing Chariots of Fire for him as he crossed the line. So he's a rather precocious little kid, but he's a real, hopefully he's going to be the pin-up boy to show that this is going to make a difference. Now, there are outliers. You get some boys with muscular dystrophy that are doing better than they should. But the thing is, we've shown that Billy is making the protein, making the missing protein. There's more of the missing protein after treatment. So you've got, so this is consistent with the disease being stabilised. Now, on spinal muscular atrophy, this is the leading genetic cause of infant mortality, as you're all aware. I probably don't even have to go through this. The human disease is caused because we have a backup copy. We've got a backup copy called SMN2. It codes for the same exactly the same protein, but there's a subtle variation in exon 7 that means it's not processed properly. So SMN2 makes a non-functional product, generally. And the thing is, we know that additional copies of SMN2 will reduce severity, and this is where the type 2s and the type 3s come in. If you've got just one copy, if you don't have a SMN1 due to the deletion, but you've got a couple of copies of SMN2, you'll typically have the severe form of the disease. Not always. Again, there are outliers. There are modifiers of SMN expression. This is what one of my PhD students, Ayantha Patel, is looking at. I'll talk about that in a second. It varies between us. And when you've got a functional SMN1, SMN2 doesn't matter at all. But the numbers vary. And you'll have between 2 and 8. So it's a duplicated copy, and sometimes you'll have 2 or 3, 4 copies on one allele. You might have no SMN1, but you could have 3 or 4 copies of SMN2. So the numbers vary. And then it depends what's happening on the other chromosome, the other allele. From mum might have no SMN1 and 5 copies of SMN2. Dad might have no SMN1 on one chromosome and 2 copies. So you'd have 7 copies all up. So it varies between the individuals, between the individual families. And what they've shown is if you have 8 copies of SMN2, you don't need SMN1 at all. So if you had an allele with no SMN1, 4 copies, and then the same on the other, from the other, the mother or father, 8 copies of SMN2, no symptoms. So, but the thing is, if you've got only 2 copies and 2 copies of SMN2, you've got 4, that's better than nothing. And I'm not sure if I've actually got that slide in, I might have taken it out, but the more copies of SMN2 you've got, the better. Maybe it'll, so basically, by changing SMN2 expression, we've got a potential therapy, and this is the basis of it. So on this slide, which I shamelessly took out of one of the a recent science paper, here's the normal SMN1 gene, and 6, 7, and 8 are spliced together to make a functional product, and here we've got our healthy motor neuron. The SMN2 gene, there's a single base change in exon 7. So here it's a C, here it's a T. And because of that T, most of the transcripts are missing exon 7. You've got 6 to 8, non-functional product, and you've got spinal muscular atrophy. But there's just enough of the SMN2 making a normal product to keep the kids alive. Now, when they started making animal models and they knocked out the SMN1 gene, or the SMN gene, all the animals, it was embryonic lethal. So this is an absolutely fundamentally important protein. And the reason we have the disease is these copies, the variable number of copies of SMN2 that give just a trickle of the normal product to keep the kids alive.
Do the amount of copies copies of SMN2 differentiate between the different types? Are there specific?
Generally, the more copies of SMN2 you have, the milder the disease. Having said that, there are outliers. I don't have the exact figures here, I can look it up afterwards, but there are some type 2, type 3 patients that only have 2 copies of SMN2, but for some reason they've got some other genes that modify their expression so that those SMN2s work better than normal. So, and again, that would be another therapy. If you can find out what those modifiers are, we can enhance the treatment. As I said, we're a complex interplay of 25,000 different genes and then all those variants. So you can't lump everything together. We don't fit into precise boxes. So in SMA, good it's come through, you've got just the SMN gene. We've had a deletion of both copies of SMN1, so most of the products are non-functional, and there's just a trace coming through, and hopefully you can see it. I sort of probably put the transparency too high. There's just a residual amount of the normal product. So there are ways to try and treat it using small molecules, and there are a number of trials going on at the moment. I'm not familiar with these, but AstraZeneca have got a drug, sodium phenylbutyrate, valproic acid, hydroxyurea, and what's this one is Zurulene. These are all splice modifying compounds that hopefully will shift the processing so that instead of making the non-functional product, we get more of the normal product. So these are small molecules. There are, as I said, several trials going at the moment. Some are being recruited, recruiting patients, and time will tell if that makes a difference. I think in most situations here, they're looking at the type 2, type 3 patients. Now there's gene therapy. I'm not going to go into detail here. With gene therapy, this is the classic gene therapy approach of taking a functional gene, you put it into a virus that you've specially modified to deliver the payload, and it's a self-complementary AAV virus. There's a particular vector and serotypes. There's lots of different types of AAVs. This one has been tailored to deliver the SMN payload into the cells. Now the trouble is with this therapy, with sort of any gene therapy, is you're using a virus to infect cells. You've got to make sure that that virus doesn't mount an immune response. You get a cold, your body fights it. So this is work being done by Jerry Mendell in Nationwide Children's Hospital, and I forgot to mention that Jerry Mendell is also doing the Duchenne trials. He's the principal investigator. So this is in very, very good hands. It's Brian Casper, I think, is involved in developing or constructing this vector. So the Nationwide Children's Hospital, the gene therapy unit there, is really right at the forefront of all this research. So the idea is to introduce a new SMN gene to make the SMN protein and a healthy motor neuron. The work that we've been...
Sorry, Steve. Would that require, the gene therapy, does that require just constant uptake?
It depends. You probably can't. You'd have to use a different vector each time because your body would recognize that as foreign. So that it could work for a few years. And then they'd have to tweak this around to make it slightly different and then re-administer it later. The beauty of gene therapy, if you've got a vector that's going to get in there, I mean, like herpes. Herpes is for life. And that's what you really want in this situation. You want a vector that's going to deliver the payload, not mount an immune response, and keep expressing the gene.
So that's a one-off?
Potentially, it could be a one-off. But the gene therapy has been around for a long time, and there were a lot of promises in the beginning, but now we've realised that we're a bit more complicated than we originally thought. So they're now making very, very sophisticated vectors that will sort of deliver the payload safely and sustain expression. the work that we've been doing is on using the antisense oligonucleotides, these genetic band-aids, our spanners. So there's other groups doing this as well, but the idea is that we want to try and interfere with the processing so that we reinforce recognition of SMN7, exon 7, so that we make the mature product. So going back to this slide, there's not going to be a test at the end of it, but this is an earlier slide where in Duchenne, we were taking the mutation and taking it out. What we're doing here is we're trying to reinforce the mutation and put it back into the message. Exon 7 is missing. We're looking at ways to try and reinsert exon 7 in the message. So it's directly the opposite of what we're doing with muscular dystrophy. So hence, we go back to this slide. SMN has a defect here. The splicing machinery doesn't recognize it because of this silencer just downstream. So by putting a spanner onto our silencer, what we're going to do is start to get the splicing machinery to recognize exon 7 so that it becomes processed normally. So as I said, this is directly the opposite of what we're doing, and it's just a case of redesigning these spanners. It's still the same chemistry, the same principles of changing splicing, but it's for a specific application. I don't know if I'll go through this in detail, but this is the normal expression. This is in patient fibroblasts of SMN2. Now in spinal muscular atrophy, you've got this product. This is the normal product, and here's the defective product missing exon 7. So what we want is drugs that will change the expression, get rid of this bottom band and make more of the top band. And there are we've found different ways of doing it. When we put the Band-Aid on the silencer, you've changed the ratio. There's more of the top band than the bottom band. And some work that Callan Trimidopant, one of my PhD students, had done, has found another region down here that put the entire intron in. So this product here would still be functional. It should still code for a protein. That's just changing the actual gene message. And then when you actually look at the, I'm not sure you get to see this, these little green spots make people like me very excited because these are gems. And this is accumulation of the SMN protein in the cells. So we're actually painting the SMN protein in the cells. So this is a normal individual. This is from an SMA fibroblast. There's no green spots. And this is a treated SMA fibroblast. And you can see that some of the green spots are reappearing. So our Band-Aid is changing the expression of the SMN2 here and making a functional product. And working with Arthur Burgess in Columbus, Ohio, who also, Columbus, Ohio, as I said, this is a real hub. This is where Jerry Mendel is. And Arthur is an old friend. We designed a compound that was injected into the mouse model, untreated. Arthur's mice die, SMA mice die at day 15. A single intrathecal administration, no, it's intracerebroventricular administration, into the CNS. And this animal survived to 161 days from one injection. So this is very promising. I sort of tend to offend a few people by saying, if you have to use statistics to show something's working, it's not working well enough. I like sort of the more clear-cut type experiments here, where you've got a dramatic change in survival. And since this paper came out, we've further improved the potency. We've designed a slightly different compound, and all the treated animals in this study survived beyond 200 days. So that was from one injection. So again, it's giving us great hope that we can change the expression. So on that, just the summary, what we're doing is alternative splicing. We're doing therapeutic alternative splicing. We're redirecting a natural process. So in muscular dystrophy, we take an exon out that causes the disease. In spinal muscular atrophy, we're trying to put an exon back in to restore function. So this is going to be tailored. These are tailored drugs for personalised treatments, and we hope to make these applicable to many different conditions. There are a lot of challenges ahead. We're looking at basically treating the untreatable. You have to know the mutation. You have to know what gene's involved, what the spelling error is, so that you can design the treatment. These are rare diseases, but you still have to know where the patient is, where the individual is, the natural progression of the disease. So we have to build up almost a longitudinal history of how the individual's tracking. And then when you've got a rare disease, doing clinical trials becomes a challenge. You can't use the standard 1,000 people normal healthy volunteers receiving the drug, 1,000 people not receiving the drug, and what are the differences. Safety and evaluation with small studies is really very challenging. And again, when you have got a limited population of, okay, boys with muscular dystrophy or people with SMA, you don't want to be in a placebo group. You know what's going to happen. The natural history is pretty established. And it's going to take time to show that the disease is being changed. The first systemic study for muscular dystrophy was 12 weeks, and they didn't see a difference between treated and untreated. And in hindsight, that's bleeding obvious. When you're renovating a house and you slap on a coat of paint, you might make it look a little bit better, but you're not changing the structural integrity of the house. It takes a lot more to lay down the foundations, replace the girders, and so on. And the safety and sustainability of the drug production. These are the morpholino drugs that I've got great hope for, great faith in. They can only make small amounts of this at the moment. The company we're dealing with in Boston, Sarepta Therapeutics, are in the process of scaling up production. But if this works, as I hope, they're going to have to scale up to astronomical proportions. And then on the shameless hype that I'm trying to sort of push into this is that we've got a demographic time bomb ticking. We're all getting older. No one's getting any younger. And the over 65s are going to increase to 25% by 2050. We're going to have a lot more Alzheimer's, Parkinson's, and the aged care. The cost of this is going to become astronomical. If we can use the rare diseases, the single mutations, and like with Duchenne, we've changed it one way, with SMA we changed it another way. If we can come up with some treatments for Alzheimer's, that's going to get the pharmaceutical companies very, very engaged in this type of technology. I was actually giving a talk on Thursday in Perth for some businessmen, and I sort of finished off with, is ageing a disease? And there is some evidence that there is certain proteins that are increased in ageing. So if you could stop those proteins from accumulating in your body, you might be able to slow down the ageing process. So this would get a lot of people involved and engaged in this type of technology. This really is hype. It's down the track. But again, only a few people know SMA. Everyone knows ageing. But if we have to use these rare diseases as guinea pigs to show that we can change the course of a disease, we start with the serious genetic diseases, and then we can extend it to the more frivolous ones like ageing. So I'd just like to acknowledge the group. This is Iantha, and this is Lauren. The money is going towards consumables for these guys to continue their experiments. Whenever we make these little genetic band-aids, they're expensive, and the actual cost of doing these experiments, to grow patient cells in a dish, to add our band-aids and do the molecular testing is expensive. And Callum Chai, Mitapant, I don't want to add up how much he cost. He was an absolute dynamo in the lab. He did so much work. We had to generate a lot of reagents for him to do the testing. But he set a fantastic groundwork, and he's actually Iantha's co-supervisor with me and Sue Fletcher. So they've got a great bunch of people working in the lab. This is an old out of date. That's some of the people in the lab. And just like to acknowledge the international support here and funding from various sources. And again, SMA Australia gave us the pilot money to get this work started. And from there, we were able to apply for an AFM grant from SMA Europe and we now have patents in place that have to be sort of renewed in about 2 weeks. So on that, I'll thank you, and I'm happy to answer any questions. Thank you. 39 minutes. 39 minutes. And 30 seconds. You said about 40, so I surprised myself. Any questions?
So how close is everything to using the splicing in clinical trials?
Look, I didn't mention it, but there's another company called ISIS. They'll probably have to change their name soon. They're based in they're based in California, in Carlsbad. They've got a similar technology, and they actually have proprietary rights on the target that we found. So they're actually starting clinical trials. They're recruiting, and you know they've done some nice work. So that is a possibility. I'm biased towards the morpholinos. I think they'll probably turn out to be better. But having said that, these are the first generation drugs, whether it's the ISIS drug or the Sarepta drug, the Sareptasol. Sarepta are designing different chemistries, different. Think of these as sort of like spanners with different tags to infiltrate the machinery. A bit flexible so they can sneak into the machinery a bit better. Something that's even stronger. So there's lots of ways we can improve this.
At this point, does it look like they're all spinal injections?
No, actually we're looking at compounds that would cross the blood-brain barrier. So it'd be a systemic administration. Now this SMA, it's the motor neurons that die first, but the SMN protein is essential for everything. So right throughout the body. If there's no point just correcting the CNS, the motor neurons there, and then having liver cells or kidney cells die or something like that. It's got to be a systemic treatment. So most likely it will be systemic administration.
So you're saying, Steve, you can get past the blood-brain?
There are ways of doing it, yeah.
That's what we've been told is the major.
It's a challenge. One of the things. You can have, just imagine that the spanner with a little homing tag that drags the spanner through the cell across the membrane. Because I'm talking about just getting a spanner and just dropping it into the machinery. You've got to inject it. It has to be taken up by the cell, so it's got to cross the cell membrane. Then it has to be transported through the cell. Then it's got to cross the nuclear membrane. Then it has to get in there. So there's a lot of travelling. So there's ways of facilitating, making that a little bit faster and more efficient. And we're looking at that. Other groups are looking at that as well. So there's a lot of ways. It's still a long way to go, but we've shown that in the animal models we can make a difference, and it's going to be a case of advancing as far as you can, checking the consequences, and then modifying the treatment accordingly.
What is the prospects for people that are already dealing with this? So will it be effective for those people that are...
We don't know. Just really don't know. With the Duchenne work, again, this is a different condition. It stabilises the disease. Possibly with the type 2 and type 3, it might stabilise it. We might be able to improve. I just don't know. The other thing is that if this is used in combination with other factors, neurotrophic factors that improve or restore, say, some nerve growth, and then stabilise the nerves as well. It's a combination. I mean, you could be combining cell therapies and exon skipping. The small molecule compounds, whether it's the hydroxyurea or valproate or something like that, and exon skipping. You don't have to use just one treatment. You mix and match. Like taking an antibiotic. You don't take single antibiotics for the severe cases. You take a cocktail of drugs.
Has there been a lot of testing on the valproate?
I'm not familiar with it. The valproate's been used in a number of different diseases. So I don't want to. I can't comment on it.
They're closing. She's been on bed break since 2.
Okay.
We've seen, as you said, stabilisation.
Okay.
Yeah, it's, I guess lucky. They say it doesn't work in some as well.
That's that's good. And again, it's anything that can buy time is going to be important. There's a lot of excitement about the gene therapy trials that are being planned because if you can restore massive expression,